Hi there, my name is Laura Upton and I'm an early stage researcher in the SimpleSci consortium. SimpleSci is a collection of leading European research institutions and partner organisations that have banded together to support 14 neuroscience PhD candidates or early stage researchers. Together we're tackling a common goal to further the body of research into synaptic dysfunction in neuropsychiatric disorders, such as autism spectrum disorder. Autism spectrum disorder is a disorder that can involve both neurological and psychiatric symptoms. It is characterized by an impairment in communication and social interactions, as well as restricted interests and repetitive behaviors. Autism is common, affecting roughly 1% of the general population. Autism spectrum disorder gives rise to varying levels of disability and is often associated with other problems, such as cognitive impairment, epilepsy, and other psychiatric diseases. As such, autism spectrum disorder can profoundly affect the lives of patients and of their families. There is a strong contribution of genetic factors in the origins of autism, meaning the genetics of autism is an important domain for autism research. Now for a quick genetics lesson. DNA is found in almost every cell of our body and is the molecule which makes up all of our genes. Genes are the biological blueprints of our proteins, which are large molecules that are the building blocks of our human body. The entire set of genes that makes up the human body is called the genome. A normal gene will encode a normal and functional protein. However, sometimes genes become altered, and these alterations can result in the production of non-functional or malfunctioning proteins. This type of alteration to a gene is called a genetic mutation. Over 200 genes with disease-causing genetic mutations have already been implicated in autism, but since the genetic cause of autism is known in just 30% of cases, these only represent a small fraction of the genes involved in autism. This means there are many more autism genes that are yet to be discovered. Our group is involved in large-scale genetic studies of individuals with autism spectrum disorder. The results of these studies enable us to identify mutations in genes that result in autism or other neurodevelopmental disorders. In addition to gene discovery, it is important to understand how these genetic mutations result in patients developing autism. To study how mutations in some of these genes can change the brain and behavior, our group uses genetically modified mouse models. These are mice who have had the gene of interest deliberately mutated so that the gene does not function. We can perform molecular and behavioral studies on these mice to gain a more holistic understanding of how these mutations result in autism in patients. A few years ago, our lab discovered through large-scale genetic studies that the GLRA2 gene was mutated in 3 patients with autism. The GLRA2 gene encodes the glycine receptor alpha-2 subunit protein, a protein found at or near communication points between brain cells, known as synapses. It plays an important role in synaptic and extrasynaptic communication and signaling in the brain. A series of experiments determined that the mutations detected in patients resulted in the absence of functioning glycine receptor alpha-2 subunit protein. My project aims to characterize the effect of loss of this protein using a genetic mouse model that lacks the GLRA2 gene. With this model, we can examine effects on brain structure, connectivity, and behavior, which will help us understand how mutations in this gene in these patients resulted in autism. If you would like to learn more about the projects being carried out by the SimpleSci consortium, check out our website and social media links. Thanks for watching.